Bismillah ar-Rahman ar-Rahim. I would like to express my deep thanks to Professor Muhammad Hashim, President of the National Research Center, Professor Mahmoud Bahgat, Head of the Central Laboratories, Network, and Centers of Excellence. Also, I would like to express my deep thanks to the conference unit for organizing this event. Title of my talk will be Introduction about Molecular Spectroscopy and Fluorescent Laboratory. I am Medhat Ahmed Abdel Khali Ibrahim, Professor of Applied Spectroscopy and Molecular Modeling. I'm working in the Spectroscopy Department and Head of Physics Division. I'm member of the National Committee of Physics at the Academy of Scientific Research and Technology, member of the Web of Nuclear Research at the Academy of Scientific Research and Technology. I'm going to present the members of the group, starting with Dr. Ali Okasha, Dr. Badawi Anis, Dr. Ayman Mustafa, Dr. Asma Muhammad Ismail, Dr. Abdurrahman Munaza, Dr. Muhammad Sayed, and finally Dr. Asma Abu Zaid. In this talk we have introduction, and then we speak about molecular modeling and what is available in our group to, or what we can provide with molecular modeling, and then we have two experimental facilities, Raman spectroscopy and confocal laser scanning fluorescence microscope. And then we speak a little bit about our recent developments and then the conclusion. The introduction, we have to give some outlines about what is meant by spectroscopy. So spectroscopy in general is the area of science which is concerned with the interaction of electromagnetic radiation with matter. Of course, we mean by matter the atoms and molecules. This may be in gas phase or liquid phase or solid phase, the matter, of course. The main goal of a spectroscopic method of analysis is to provide essential tools for identification of substances. But mainly in our group, we are speaking about the molecular spectroscopy. So this is a branch of spectroscopy dealing with the properties of the molecules. It is essential for understanding the molecular properties. It is used as a stepping stone to take us to the concepts of quantum mechanics. Of course, spectroscopy as well as other modern branches in physics are dealing or based on the basis of quantum mechanics and the quantum mechanical descriptions of the molecular properties. If you are going to describe the molecular structure or the molecular behavior of some molecules, we speak about diatomic molecules, which is a molecule consists of two atoms when the incident radiation is interacting with this molecule. We have a very special physical parameter called the dipole moment. If the dipole moment of this molecule is changed, then we call this molecule is infrared active. So the structure is infrared active when its dipole moment changes with the incident electromagnetic waves. But what if this structure is not infrared active? So we have to study the molecular properties through another technique. That's why we start to use Raman technique. For Raman technique, if the incident photons or the electromagnetic wave is interacting with the molecule, then a very small part of these photons will be scattered. We have three schemes of scattering. Two of them, it loses or gains some energy. So we have so-called inelastic light scattering, which is the Raman phenomena. So simply the structure is Raman active when its molecular polarizability changes during the vibration. So two techniques, infrared and Raman, are describing the molecular phenomena. But for a biological molecule, we have some kind of special care. For example, some molecular interaction, especially those of biological origin, occur in the short periods. It's something like picosecond or nanosecond, which makes time resolved very necessary to understand the biological interaction. So this time resolved technique is useful technique in a biomolecular structure, the analysis and the dynamics. We do believe that in order to describe the molecular properties of molecules, it is necessary to apply three main concepts. Quantum mechanical calculation, which is through the molecular modeling technique we have already in our laboratory, and we have also offering Raman spectroscopy and time resolved spectroscopy. So first we start with the quantum mechanical calculation, and I will not go through the basis of quantum mechanics, but I will make some kind of few elements about what is molecular modeling, which is a class of computational work which is working with the molecules, and it simulates chemical structure and reaction numerically based in full or in part on the fundamental laws of physics. Molecular modeling provides precise physical and chemical data and serves in many areas of research. If we are going to describe what we can gain or what we can calculate through molecular modeling, let's start with, for example, the molecular spectroscopy of sediment in the River Nile. For example, if we describe what is the meaning of this band, we can make many schemes describing that this band was some kind of interaction of organic and inorganic structure in the aquatic environment, and we make so many schemes describing this interaction. It seems to be that molecular modeling in the environmental research is to explain the experimental results, or even to make a mechanism of interaction to understand what's going on in the environment and so on. In other work, we can describe what's going on for the carbon nanomaterials, such as carbon nanotubes, graphene, and fluorine. We can use this class of carbon nanomaterials as sensors, because if we make a doping metal oxide semiconductor together with nanotubes, we can have sensors. Also, if we put the metal oxide or functional groups with the graphene, we can functionalize and change the electronic properties. If you are going to describe, you have some schemes of interaction, doping, decoration, substitution, and so. If you have to study the zinc oxide interaction with carbon nanotube, which technique you have to do experimentally. So through molecular modeling, we can design, then implement certain experimental facilities based on the electronic properties of the molecular modeling. We have so many schemes studied, we have published so many work, and we are expert in this area to provide some facilities to describe some interaction, not in biological or environmental, but also in the field of nanomaterials. And now let's move to the first experimental techniques that we have, which is Raman spectroscopy, which is a very, very specific technique, and this will be presented by Dr. Badawi Anis. He's expert in this field. He's got his PhD from Germany. He's working in carbon nanomaterials under extreme conditions, and he will going to present the Raman facilities available in our group.
Hello everyone. In this short video presentation, I'm going to talk about Raman microspectroscopy with some practical applications. This facility is present in the molecular spectroscopy lab in the Central Laboratories Network in the National Research Center. I talk today about why Raman microspectroscopy is a very important technique. And as we can see here in the schematic description, we have Raman microscope. In this Raman microscope, we can use laser source as excitation source, and then using some focusing optics, we can focus the laser beam on the specific point on the surface of our sample. Since the reflected Raman spectrum can be go directly using some kind of fiber optics to see spectrometers, the spectrograph, using some different kind of grating, so we can distinguish between several Raman peaks. Then we can send this spectra to CT spectrograph or CT detector. Using Raman software, we can distinguish between several peaks and several contributions from our sample. In our lab we have this nice instrument which is VTech 300 RA from Germany technology. Using this instrument we can record or can measure single point spectrum with high resolution. Also we can measure Raman spectral imaging for surface of for a specific sample, and also we can perform depth profiling. With our microscope we have two excitation wavelengths, 532 and 785 nanometer. We can change the laser power from 100 microwatt to 50 milliwatt. Okay, and somehow Raman spectroscopy is similar to FTIR spectroscopy. In FTIR spectroscopy, the absorption of the incident light occurs when there is a change in dipole moment, but here we have a change in polarization of molecule. So if we excite a molecule with green laser, for example, the molecule can excite to visual state, from ground vibrational state to a visual state. Then this photon return back again after inelastic scattering. So if return back to different state with lower energy than the incident photon, so we have stock scattering or we have anti-stock scattering. Also if the photon can excite to virtual state and return back again with elastic scattering, so we have realized scattering. Okay, why Raman spectroscopy is very important technique. Here we can see some practical applications for our microscope. Here we can distinguish between Raman and the FTIR. Here we have some organic compounds like iodine. So we see FTIR spectrum. We can see very nice spectrum. It contains many peaks, many sharp peaks and overlapped peaks and shoulders, so on. So it's somehow a complicated spectrum. But Raman spectrum for the same compound contain few peaks and resolved peaks, and so easily we can distinguish the composition of our organic compound. Here we have another example, methanol and ethanol. Methanol and ethanol are well-known organic alcohol. Here we have, in case of ethanol, we have only one peak from CH3 deformation, and here for methanol we have both from CH3 and CH2 functional group deformation. So from Raman we can distinguish easily between methanol and ethanol, which is not easy with FTIR spectroscopy. Also for chemical identity of similar compounds like this blue and red compound or red spectrum like mesamphetamine and MDMA, we can see that both compounds have more or less the same structure, except here we have two extra oxygen atoms. For this red compound we can see that sharp peaks, which is different from blue one, so we can distinguish between these two similar compounds. Also, using Raman spectroscopy, we can distinguish between different salts like magnesium sulfate and sodium sulfate. Here we can see the red one for sodium and the green one for magnesium. We can see some significant shift between these two characteristic peaks. Also using Raman spectroscopy, we can distinguish different crystal phases for the same compound. Like in case of titanium dioxide, we have anatase and rutile structure. This red one for anatase and this blue one for rutile structure. So using Raman, we can know if we have anatase or rutile, or we have a mixed phases from both rutile and anatase. Again, we can detect any change in the crystal structure for the same sample. Like if we applied extreme conditions on the sample, like pressure or like temperature, for sure we have the same structure, crystal structure, but we have some, for example, some stress on the crystal structure. So we can easily distinguish between unstressed sample and stressed sample, like in this kind of crystals, so we can find any change due to any external physical property like pressure or temperature. One of well-known and advanced material, which is graphene. Graphene is a two-dimensional material with one atomic sec. So we in this case we have monolayer graphene. We could have bilayer graphenes and 3 layers or multi-layers graphene. Using Raman spectroscopy, we can find easily how many layers we have in our sample. In case of monolayer graphene, we can see that the 2D peak is higher than G peak, and the ratio between them is 2.5. For in case of bilayer graphene, the ratio is about 0.9 or 0.8. In 3 layers, we have the ratio between 2D and G is about 0.5. Also, using Raman spectroscopy or especially microspectroscopy, we can distinguish between number of layers in molybdenum disulfide. Molybdenum disulfide is also has a hexagonal structure like graphene, but it's semiconducting material. So we can find if we have monolayer, bilayer, 3 layers, 4 layer, 5 layers, 6 layers, only to calculate the difference in position between the E2G mode and E1G mode, and in this curve we can see a relation between number of layers and the position or the difference between these two phonons. So we can, in case of monolayers, we have about 18 or 19 centimeter minus 1. Here we have about 21 in case of bilayer, till we reach to bulk molybdenum disulfide. One of the most important application of our microscope or Raman microspectroscopy in general to do mapping. So mapping, we can scan very large area for a few micrometers to millimeter area, and we can identify if we have one compound or several compounds. Here we have a complex phosphatic sample, so each color represents different structure or compound in the sample. So we can give each one different color, and we map and we have a general overview about the distribution of the sample. Again, we come back to graphene. Also, we can find, in case of graphene, if we have monolayer, bilayer, 3 layers, just we can scan large area, and calculating the ratio of 2D over G mode, we can see if we have homogeneous monolayer or bilayer or trilayer. This technique is not easy to know if we have monolayer, bilayer, 3 layers for large area, few micrometers by micrometers. Only we can know if we have monolayer, bilayer, or 3 layer using high resolution transmission electron microscope. This technique is very difficult and very sophisticated. We need to transfer graphene on very expensive copper grid, and only with high resolution microscope we can scan only 1 point, a few nanometers by nanometers, which is not representing the quality of the sample very well. But in Raman we can scan very large area and know the homogeneity of our samples. Again, using Raman microspectroscopy, we can make a spatial resolution or penetration depths. Using a different kind of lasers with different wavelengths, we can we have several penetration depths. For longer wavelengths, like in case of red laser, it penetrates more, so it can reach to lower lower depths. In this case we have silicon germanium layer, uniform silicon germanium, strained silicon. So different lasers here, we can go to different depths, and we can identify the composition of the sample or of the layers. Here another example, we have different polymeric materials in film. We have polyethylene, nylon polyethylene. So also we can distinguish this top surface is polyethylene and this middle one is nylon. Okay, finally we can conclude why Raman spectroscopy is very important technique. In general, Raman microspectroscopy is a relatively cheap technique and a non-destructive technique we can use to distinguish several materials in different phases, like liquid or solid. We don't need to do any preparation, extra preparation for our sample. We can measure our sample directly. If it is powder, if it is thin film, we can distinguish if it is different polymeric material. We have different crystal structure. We have monolayer or several layers from graphene or from molybdenum disulfide and 2D dimensional material in general. Also, we can map the surface of some thin films material to know the homogeneity of the surface, homogeneity of our sample. Also, we can perform depth profiling for many materials. Thank you very much.
Thank you, Dr. Badawi, very much. And now we speak about the confocal laser scanning fluorescence microscope, which will be presented by Dr. Ayman Mustafa. Dr. Ayman is a researcher in the spectroscopy department. He is working in the field of laser spectroscopy. Dr. Ayman, please.
A microscope is an instrument used to see objects that are too small for the naked eyes. And the science used to investigate this object is known as microscopy. The microscopy is a combination between two very important items. First one, resolution. Second one is contrast. Resolution meaning to seeing smaller and smaller things, while contrast meaning seeing what you want to see. To understand the basics of microscopy, let's go to Jablonowski energy diagram. When the light makes an interaction with certain substance, and this substance absorbs a photon from this light, the electron was excited from the ground state to higher level of the excited state. At this time, the electron can relax via collision or vibration or emitting another photon. In the case the electron relaxes before emitting another photon, there is fluorescence process, and at this time is known as fluorescence microscopy, and this sample known as fluorophore. If you are interested with biological samples like plant, human tissue, animal, DNA, at this time fluorescence microscopy is a good choice for you. There are different microscopes could be used for fluorescence microscopy, as simple microscope, which has only one lens, compound microscope, which has two sets of lenses, and it allows to make a magnification 100 to 1000 times larger than they really are, or electron microscope, which can magnify objects up to thousands times. The main process of the microscope was based on using a multi-wavelength source, could be arc lamp, mercury lamp, polychromatic, and then passing through excitation filter and objects, and then making an interaction with the used sample to make an excitation of this sample and to produce a fluorescence which was reflected by dichroic filter and then go to use detector. This type of microscope is known as wide field microscope, but this microscope has a drawback in a high magnification value, as the image seems to be blurry. So we need to enhance the contrast. For that, the scientists find an intelligent idea based on added a pinhole in the structure of the wide field microscope. At this time, if the whole sample illuminated by scanning single wavelength laser, only the light from of the focal plane is passing through the pinhole to the detector. This type of microscope is known as confocal microscope. So the confocal microscope was used to enhance the image, especially at the high magnification value. Also, it used to do time-lapse movie with the confocal, as shown in this number of images, which was taken to study at the z direction beside the xy scale. In some cases, we need to use IR laser to penetrate deeper into the tissue, rather than shorter wavelengths. From this relation, the IR laser has higher wavelengths and a low value of energy, which wasn't sufficient to make an excitation of electron from the ground state to the excited state. In this case, we use two photons excited only occur at focal plane to make an excitation process and to produce the following spectrum. Now we will go to study the application of fluorescence lifetime microscopy with different methods, as fluorescence lifetime imaging microscopy (FLIM), stimulated emission depletion microscopy (STED), fluorescence resonance energy transfer (FRET), fluorescence correlation spectroscopy (FCS). Let's start with fluorescence lifetime imaging microscopy, which is known as. It used to measure the lifetime between excitation and emission, as every fluorophore has unique nature lifetime, which could be changed by the environment, as ion concentration, oxygen concentration, pH, hydrophobicity, or protein-protein interaction. This method based on excitation of many electrons at the same time, and count the different time when they are falling back down. Then measure the lifetime, which represents as half of all electrons are falling back. At this time, we are producing exponential curve as shown in the following spectra. The second technique is stimulated emission depletion microscopy, or STED. It is a super resolution microscopy technique that used to get a high resolution image at the molecular level. But the problem appeared here when we imaging a biomolecule when the scale from 1 to 50 nanometer, like viruses or microtubules, the image was appeared as a blurry. Therefore, the confocal microscope must be developed. The scientists found that the solution must be focused on decreasing the excitation spot of laser source. At this time, we can get imaging just of the individual molecule to get just its fluorescence without any effect of the neighbor molecules. However, it is impossible to achieve a very small excitation spot reached to 20 nanometer or below. This phenomenon known as point spread function, which is stated that the smallest spot that a convex lens can focus cannot be less than the wavelength of light. As we have a laser source of 500 nanometer, the minimum spot size cannot be below than 500 nanometer. At this time, the scientists find a brilliant idea based on using stimulated emission process. As when the light makes an interaction with a substance and the substance absorbs its photon, the electron was excited from the ground state to excited state. And before the electron go back to the ground state, it was re-excited again with another wavelength. At this time, the fluorescence was emitted but with a different wavelength, lower one. This process known as stimulated emission process. This technique contains also an emission filter to allow only one wavelength to pass and block the others. Therefore, this technique was based on using two types of laser source. First one is used to make an excitation of electron from ground state to excited state, and another one was used to make a re-excited of the excited electron. And the shape of the image by using two types of laser source in this method seems to be donuts, and at this time we can image the molecule below than 20 nanometer scale. By using this technique, we enhance the contrast and the resolution of the image as we see in this studied sample. At this time, we don't need a computational reconstruction as we doing before. The third technique is known as fluorescence resonance energy transfer, or FRET. It's used to investigate molecular interaction. It based on a closed acceptor molecule can take excitation energy from the donor. At this time, we can study the energy transfer without no emission by using the following equation, where R naught is transfer efficiency density. As we see, when we are becoming too closer, the overlap is increased, and when we are out of the distance, the overlapping will disappear. At this time, we can detect the energy transfer as shown in the following figure. The fourth technique is the fluorescence correlation spectroscopy, or FCS technique. FCS is a high-resolution spatial and temporal analysis of very low concentration of biomolecules. This is done by measuring the spontaneous intensity fluctuation caused by deviation of the system from equilibrium. It based on detection of single molecule excited to fluorescence in diffraction-limited confocal volume element. The produced spectrum was appeared as shown in this figure. These fluctuations are caused by diffusion of fluorescence molecules through the cavity or just a change in the fluorescence over time. The correlation functions are used to obtain quantity information about diffusion coefficient, hydrodynamic radii, average concentration, kinetic chemical reaction rate, singlet triplet dynamic, and number of molecules.
Thank you very much, Dr. Ayman. Now let's move to the recent development in our group. Last year we granted Egyptian patent for gas chamber for testing the efficiency of volatile gas sensors, and last month we have finished a national initiative funded by Academy of Scientific Research and Technology entitled Cost-Effective Design and Implementation of Microsphere Production for Wastewater Treatment. This work was in collaboration with the Spectroscopy Department and Water Pollution Department. By the end of the last year, we participated in the Cairo International Exhibition for Innovation, and we got a golden medal. Recently, last month, we participated in the fourth forum for the research output organized by Academy of Scientific Research and Technology. We present something there. We got the I got the STAT prize in technological science also last year, and I was really lucky to work among this wonderful group. We make a contract with the Academy of Scientific Research and Technology for a project entitled Multidisciplinary Analysis for Soil Sediment in Egypt. We are now preparing an atlas for the molecular structure of soil and sediment. We are going to collaborate with other groups working in the field of agriculture because we have some deficiency in the structure of soil, and then we can prepare with the help of them some kind of organic fertilizer from green resources. Also, we are willing to collaborate in not only in the National Research Center, but also with other colleagues from other research centers and from other universities. And now we come to the conclusion. We are really willing for collaboration, and we are also offering both theoretical and experimental facilities to understand the molecular phenomena in many fields of research, not limited to environmental or biological research, but also for nanomaterials, and we can collaborate to make some mechanisms and some descriptions for the molecular interactions. Thank you very much, and I would like once more to thank Dr. Mahmoud Bahgat for his continuous support. I would like to thank all the team members, and we are here for your questions, and we are willing to answer them if we can. Thank you. شكرا جزيلا والسلام عليكم.